This is Module A2, Objective 2, and this objective deals with the specifics behind carbohydrate metabolism. We're going to look at the basic steps in glycolysis, the TCA cycle, and the electron transport system. If we were to look at just a quick overview of carbohydrate metabolism, we would note that a molecule of glucose plus oxygen will yield carbon dioxide, remember this is waste, water, and a tremendous amount of energy. If we consider glucose metabolism, we find that one glucose molecule is broken down in small steps. And ultimately, the energy from those that breakdown is converted and stored in the form of ATP. And we begin glucose metabolism with the process of glycolysis. And if we were to break down this word, glyco refers to sugar, and lysis refers to breakdown. And glycolysis occurs in the cytosol of the cell. Glycolysis proceeds without oxygen, or without the need of oxygen, and this is referred to as an anaerobic reaction, a reaction in which oxygen is not required. And we can contrast that with aerobic reactions, and we'll look at aerobic reactions later on. Glycolysis begins with a 6-carbon glucose molecule. And again, if we were to try to place ourselves using this figure, here we see the plasma membrane. We have the interstitial fluid outside with the cytoplasm inside. And we start out with one molecule of glucose. Glucose is often taken in dietarily, and then it passes through the cardiovascular system, or via the cardiovascular system, and is delivered to every cell in the body. Glucose is taken into the cell, and we get the process of glycolysis. And glycolysis consists of 7 steps. Now, there is a tremendous amount of detail, and if this were a biochemistry course, I would expect you to know all of the details. But what I'm really interested in is that you know what we start with, what we end up with, and maybe a few specifics in between. So we start off with a glucose molecule, which is a 6-carbon molecule. And what we note, or what we see, is that that glucose molecule is being altered as we move down through the steps of glycolysis. And at every step, we have an enzyme that is actually it's promoting this alteration. But if we look at glycolysis overall, what we have is we start with one molecule of glucose. We end up with 2 molecules of pyruvic acid. In terms of energy, we end up with a net gain of 2 ATP. And I just almost highlighted right through that word. 2 ATP. So, what are we going to remember? We start off with glucose. It enters the cell, and within the cytoplasm, or the cytosol, we have the steps of glycolysis. And ultimately, we end up with pyruvic acid, 2 molecules of pyruvic acid, and 2 ATP molecules. What is needed for glycolysis? Obviously, we need glucose molecules. We need all of the enzymes that facilitate the alterations or the changings between all of these 7 steps. There's a specific enzyme that converts glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 1,6-bisphosphate. Each one of these steps require a different enzyme. We need ATP, ADP, and inorganic phosphates in order ADP and inorganic phosphates are used to create ATP. And we also need a coenzyme known as NAD. And NAD is a very, stands for a very long name. I'll spell it out for those who are interested. It's nicotinamide adenine dinucleotide. Let me make sure I spell this right. And obviously you're not required to remember that. For now, just know that it's a coenzyme, and it becomes very important later on when we look at energy production. So one molecule of glucose is broken down anaerobically without oxygen to produce 2 molecules of pyruvic acid, and ultimately we get 2 molecules of ATP. All right. Those 2 molecules of pyruvic acid will now enter the mitochondrion. And in the presence of oxygen, we're going to undergo, we're going to further carbohydrate metabolism. Let's quickly take a look at the structure of the mitochondrion, and I think this next slide will actually show that. The mitochondria is a double membraned organelle. We have an outer membrane and an inner membrane that has increased surface area. We see that there's all these involutions that run through the inside. And what that does is it allows an increased surface area for the placement of enzymes. And these are the enzymes that are actually going to continue metabolizing this carbohydrate, or now these pyruvic acids. So the outer membrane has very large diameter pores, and it's permeable to ions and some small organic molecules such as pyruvic acid. The inner membrane contains the carrier protein, and it's the inner membrane that actually moves the pyruvic acid into the matrix of the mitochondria. We didn't define the matrix. Here's the matrix, and the matrix is this space that is created within the inner membrane. And between the inner membrane and the outer membrane, we have the intermembrane space. This is a flow diagram of the TCA. This is the tricarboxylic acid cycle. And I don't think, let me just write that down. It's carboxylic. Many of you might remember this as being the Krebs cycle, but I think most textbooks are going to the tricarboxylic acid cycle. It's easier to spell, I'm sure. The whole function of the tricarboxylic acid cycle is ultimately to remove hydrogen atoms from organic molecules. Okay, main function, remove hydrogen atoms from organic molecules and transfer those hydrogen atoms to coenzymes. And we mentioned coenzyme NAD, and I'm going to introduce the other coenzyme, which is right here, FAD. So again, the function of these coenzymes is to basically pluck hydrogen atoms off of these organic molecules. And we see that the TCA cycle, we start off with pyruvic acid. This moves into the mitochondria, and it's quickly converted into acetyl-CoA. On and on. As we move around this cycle, you get a conversion. And what we ultimately end up with is a little bit of ATP right off the bat. We end up with a tremendous amount of NADH. This is one of these coenzymes, NAD, that has plucked a hydrogen atom off of this organic molecule. And we end up with FADH2, which again has plucked hydrogen atoms off of these organic molecules. The coenzymes, FADH2 and NADH, all end up, if you follow the arrow scheme, and enter what is known as the electron transport system. Within the electron transport system, we have a process that is called oxidative phosphorylation taking place. And ultimately, this is where 90% of the ATP that's utilized by the cells is produced. We note that oxidative phosphorylation occurs within the mitochondria, and it requires the coenzymes, the NADH and the FADH2, and it's aerobic, so it needs oxygen. This is kind of an overview of the electron transport system. What we find is that on the inner membrane, we have a number of proteins, and they all have individual names, but for now I think that we're just going to identify them as proteins. And we see that the NADH is going to deliver those hydrogen atoms to this series of proteins. And that hydrogen atom is going to start being passed from protein to protein. While it's being passed early in this system, we note that the hydrogen ion is released. If we look at a hydrogen atom, we note that it has one proton and one neutron in the nucleus, and then it has one electron, signified by an E minus, orbiting that nucleus. As the hydrogen atom is being passed from protein to protein, the nucleus of that hydrogen atom is lost, and that's what is shown right here. When you remove the nucleus of the hydrogen atom, you end up with the hydrogen ion. And what continues along the path is the electron. Ultimately, the electron is going to react with the oxygen, and it's going to produce water. Okay, so that's what happens ultimately with that with that electron. The hydrogen ions that are lost early in this process are going to be pumped across the inner membrane into the intermembrane space. And ultimately, this will create a concentration gradient. Where you have a whole bunch of hydrogen ions in the intermembrane space and very few hydrogen ions within the mitochondrial matrix. So we have the production or the creation of a concentration gradient. And as we saw in the cell physiology lab, concentration gradients is a tremendous source of potential energy. Let me forward one slide and look at the electron transport from a different view. This actually is 25-5A, and it kind of goes through the steps. We start out with a substrate, that organic molecule in the TCA cycle, and the NAD and FAD have stripped hydrogen off of that substrate. It is then carried to the electron transport, and these are the proteins of the electron transport chain. So the hydrogen atoms are delivered, and they start being passed from one protein to the next, all the way down the chain. At some point, and here it's step 3, hydrogen ion, or the nucleus of the hydrogen atom, is lost. And what continues is the electrons. And the electrons are going to be passed from one protein to another until those electrons combine with oxygen to form water. As this is happening, again, we have pumps, hydrogen ion pumps, that are creating a concentration gradient in the intermembrane space. This is the intermembrane space. This would be the matrix of the mitochondrion. So these pumps are pumping hydrogen ions into the intermembrane space, creating a huge concentration gradient. And if you remember from lab, the idea of equilibrium, what this system wants is the same number of hydrogen ions on one side of the membrane as the other. So there's a tremendous amount of potential energy located here. And I'm going to back up so we can look at this diagram again. And what we have here is an ion channel, a hydrogen ion channel. This is a channel, a protein channel, that actually allows hydrogen ions access to the matrix. And since the potential energy, or the concentration gradient, has been created, these hydrogens are going to eagerly find their way through this channel back towards the matrix. And as the hydrogen ions pass through the channel, you get the production of ATP. This hydrogen ion channel is associated with ATP synthase, and the ATP synthase produces ATP. And this, folks, is where 90% of the energy used by the body is produced, right here. So, the electron transport system does not produce ATP directly. What the electron transport produces is water. A byproduct of the electron transport system is a concentration gradient across the inner mitochondrial membrane. And this is a situation, again, where you have the inner membrane and hydrogen ions are being pumped into the intermembrane space, creating a concentration gradient. A lot of hydrogen ions on one side, very few on the other. This system wants to reach a state of equilibrium. It wants the same number of hydrogen ions on one side as the other. And we have in this membrane a channel, a hydrogen ion channel, that allows hydrogen ions access back across the membrane into the matrix of the mitochondria. Associated with this channel is an enzyme, a protein known as ATP synthase. And this is the protein that allows ADP plus inorganic phosphate to form ATP. This slide notes the ion pumps and the ion channels. I've already mentioned them twice, so I'm not going to do it again. You can read it on your own. For one NAD, the energy of one electron pair removed from the substrate, and this is just referring to those hydrogen atoms that are plucked off the substrate during the TCA cycle by NAD, that one electron pair will pump 6 hydrogen ions into the intermembrane space to create that gradient, and it's going to give rise to 3 molecules of ATP. So what I've always memorized is NAD is going to give rise to 3 ATP. In terms of the FAD, 4 hydrogen ions are pumped into the intermembrane space, and you get the generation of 2 molecules of ATP. So FAD equals 2 ATP. So for every coenzyme NAD that, or NADH ultimately, once those hydrogen atoms have been plucked, you get the production of 3 ATP molecules. And for every coenzyme FADH2, you're going to get 2 ATP molecules produced. We begin with glucose, we end with carbon dioxide and water, and again, this is how we generate ATP. This is a summary of the entire process, starting in the cytosol or cytoplasm with glycolysis. Remember, the net gain in glycolysis is 2 ATP. The end product of glycolysis is pyruvic acid, which then enters into the mitochondria. Here's your mitochondria. And those molecules are going to enter the TCA cycle. The TCA cycle is going to produce NADHs and FADH2s. And these 2 coenzymes are going to enter into the electron transport, where you get the production of water and that little byproduct of the concentration gradient that ultimately yields the rest of the ATP. 1 molecule of glucose will give the cell a net gain of 36 molecules of ATP via glycolysis, where you get 2. The TCA cycle, within the TCA cycle, you get the generation of a few ATPs, and then ultimately the ETS, the electron transport system, is going to produce the rest. That is carbohydrate metabolism in probably 20 minutes.